All right, so this is the first link. Find it on page 2 of the lab handout. Doing DNA isolation. All right, start lab. In this experiment, oh, cute. In this experiment, you need to isolate some DNA from a human test subject. Why, you may ask? Do you need human DNA? Press next. Oh, how exciting. All these effects. Genetic testing. Ooh, body identification. Sounds important. Analysis of forensic evidence. Who was killed on this chair? Scientists isolate DNA for a variety of reasons, some of which include genetic testing, body identification, analysis of forensic evidence. Next, to continue. DNA extraction is typically the first step in a longer laboratory process. DNA extraction is an important part of that process because the DNA first needs to be purified away from proteins and other cellular contaminants. We need cells because that's where the DNA is. Inside almost every cell in our bodies is a nucleus, and inside of each nucleus is about 2 meters of DNA. Yep, 6 feet. Oh, there's the DNA. Ooh, the histones. Yeah, histone proteins. DNA is wound around those guys. And here's the DNA. So where do we begin? First, we need to collect some cells from our test subject. Ooh, oh, this poor unwitting dude. Oh my God. The skin on the inside of our mouths loses thousands of cells every day. These are the cells we're after. My God, I love these sound effects. Let's get started. The steps you follow to purify DNA from a cheek swab are shown below. Step 1, collect cheek cells. Okay, we did that. Step 2, burst cells open to release DNA. Step 3, separate DNA from proteins and debris. Step 4, isolate concentrated DNA. Press next to continue. Here are the materials and equipment you will need to purify DNA from cheek cells. Ooh. Warm water bath, centrifuge, microcentrifuge, just a little one, micropipetter, a tiny little pipetter, buckled swab, sample tube, the lysis solution, concentrated salt solution, resuspension buffer, ethanol, yum yum, isopropyl alcohol, ooh, that's rubbing alcohol. Oh my God, he's going through it again, how sad. Drab the swab over the inside of the test subject's mouth. Okay? All right. Sound effects are priceless. Now he's free. Next, you'll place the swab into an Eppendorf tube. Oh, that's just like a super tiny tube. Look closely at the swab. Eppendorf actually is the brand. Look closely and notice that it is covered with hundreds of tiny cheek cells. Inside each cheek cell is a nucleus, and inside the nucleus is DNA. Haha. Ooh, we're getting some lysis solution. The end of the swab must be cut off so you will be able to close the tube. Makes sense. Using the micropipetter, add some lysis solution to the tube. Lysis is a Greek word that means to separate. Basically, you're going to bust open them cells. Yes. Place the tube into the warm water bath. Okay? Yeah. The warm water is helping the enzymes in the lysis solution do their work. They have to be a nice warm temperature. Oh, wow. Was not expecting that. Okay. We got our DNA here. Very good, very good. The lysis solution you just added contains two important ingredients, detergent and an enzyme called proteinase K. The detergent disrupts the cell membrane and nuclear envelope, causing the cells to burst open and release their DNA. The DNA is still wrapped very tightly around proteins called histones, and the proteinase K cuts apart the histones to free the DNA. Excellent. Detergent opened up the cell for us. I don't know why I can't talk today. All right. There. Lysed cells. What we got here? Concentrated salt solution. Cells have stayed in the warm water bath long enough for the DNA to be freed from the cells. Excellent. And we have removed the swab from the tube. Add some concentrated salt solution to your tube. Okay. There we go. Salt solution is super important. Tried DNA extraction without it once. It did not work. The salt causes proteins and other cellular debris to clump together. That is correct. It also inactivates enzymes that would cut up the DNA. Place the tube into the centrifuge. Okay. In order to balance the centrifuge, a tube containing water is placed opposite your tube. Click the lid to close the centrifuge and turn it on. Yeah. That was a big mistake not adding that salt. All my DNA was chopped up from those enzymes. Inside the centrifuge, the tubes spin around at high speed. The heavy clumps of protein and cellular debris and enzymes that would otherwise chop up your DNA sink to the bottom of the tube, while the strands of DNA remain distributed throughout the liquid. Use the micropipetter to carefully remove the top liquid, which contains DNA, and place it into a clean tube. The proteins and other cellular debris stay behind. Right. There we go. What's this? Isopropanol. Ooh, rubbing alcohol. Add some isopropyl alcohol to the tube. Okay. Move that up. Get on up. Oh. Inverting the tube several times mixes the isopropyl alcohol into your DNA solution. Because DNA is not soluble, meaning it does not stay dissolved in isopropyl alcohol, it comes out of the solution. You can now see the clumped DNA with your naked eye. Press next to continue. Place the tube into the centrifuge and click on the lid to close it and turn it on. This time, after the sample spins in the centrifuge, the DNA sinks to the bottom of the tube. Yes, it does. It didn't sink to the bottom of the tube before because it wasn't clumped together. Oh, close it, yes. All right, spinning, spinning, and out it comes. Yeah. Let's get rid of that liquid. Yeah. Once the liquid is removed and the DNA is allowed to dry, you can redissolve it in the solution of your choice. You can store it in the freezer for many years, or you can move on to your next experiment. You have just extracted DNA. There it is. Try your hand at DNA extraction using common household items you can find in your kitchen. Oh, that is actually super fun. I've done that before. Okay. The end.